So this video is going to continue our conversation on the humoral response. So remember, that's focusing on B cells, which once activated and able to differentiate, are able to differentiate into plasma cells that produce antibodies. And these antibodies are little Y-shaped proteins that you can see here that are going to go out and help our immune response detect and clear the pathogen. Well, in order to do that, the antibody has to be able to bind and attach to the pathogen. And it does so by recognizing molecules associated with that pathogen. And those molecules that our body recognizes as foreign are called antigens. So this white blood cell, which is an immune cell, so this would be our plasma cell, is going to release antibodies. And notice that it is specifically attaching to this little triangular region here. That is the part of the bacteria that our body is detecting as foreign. So that's the antigen. Okay, so the antigen is part of the pathogen. All right, so we're gonna look at how the antibody and antigen interact. And so we're gonna kind of slowly zoom in on this process that you see pictured here. All right, so an antibody is a protein, okay, and it has a Y shape. And this is characteristic of proteins called immunoglobulins. So you're gonna see that IG on a couple of slides. And so when you look at this antibody, you'll notice it's a Y shape, but it's made up of 4 chains, right? These 2 dark orange, bigger chains in the middle, these 2 smaller outer chains as well. So we call these larger chains in the middle the heavy chains, and these 2 smaller chains on the outside that are kind of a tan color, we call those the light chains. Notice that we have disulfide bonds right here that are connecting the light chains to the heavy chains. And we've also got disulfide bonds here, and those are also called disulfide bridges that are connecting the 2 heavy chains together as well. Now, look at the top portions of this Y structure. You'll notice there's this little pocket here. In this case, it looks like a little rectangle. This is going to be specifically where the antigen and antibody bind. Okay, so we call this little pocket, and there's 2 of them, the antigen binding sites. Okay, and because there are going to be different antibodies for different pathogens, we refer to this region as the variable region. That's what the V stands for, because the exact shape of this area is going to vary based on what the specific target is, because the antigen on E. coli will look different than the antigen on Ebola, which looks different than the antigen on Salmonella. Okay, so remember, with our humoral response, this is part of our specific immunity. So these antibodies have a very specific target that they're after, okay? And they have to be able to bind only to that specific target. So that means this region, this variable region, especially this antigen binding site, is going to have a very specific shape. Now, we're going to talk about in a couple of slides that there are different classes of antibodies, okay, different types of antibodies that go out and do different things. But we need all of those different types of antibodies for each target. So that means all the different types of antibodies, some of them are going to be going after E. coli. The antibodies against Salmonella, we're going to have all the different types of antibodies looking after Salmonella as well, okay? And so what's going to unite each type of antibody is going to be this constant region, okay? And so all type A, or I should say class A antibodies, are going to have the same constant region. All class M antibodies will have their own constant region. So it's this constant region that says, hey, you're a class A antibody or you're a class M antibody, all right? But what's going to vary amongst the class A's is going to be which pathogen they're looking for specifically. Okay, and we're going to kind of talk more about that later when we get to that. All right, so I've mentioned that this specific little pocket here in the variable region is going to be where the antigen is going to bind. So let's zoom in on that a little bit more. So we're going to start with this top picture, and you'll notice that the little binding pocket is specifically called a paratope. Okay, so the paratope is part of that variable region. Okay, and so this is where the antigen is going to bind. But getting into more detail here, look at this picture. Do you see how large this antigen is? Right? That antigen is way too big for one little antibody to deal with. So you'll notice that there's a specific part of the antigen that's going to be able to attach to the antibody. And that specific region is called the epitope. Okay? So the epitope is the specific part of the antigen that will be recognized by the antibody. Okay? Now, to get even more detailed, bringing in more of that terminology we've used, the epitope on the antigen specifically binds to the paratope of the antibody. Okay? So I'm going to repeat that. We're going to want to write that down. The epitope of the antigen is going to bind to the paratope of the antibody. Okay? All right. Now, remember, these antibodies have a specific target that they're looking for. Okay? And so the shape of this paratope is going to have to match the shape of this epitope. And the epitope of E. coli will be different than the epitope of Salmonella, which is different than the epitope of Shigella. And so this is why, even though in our lifetime, we're going to come into contact with hundreds and hundreds, maybe even thousands and thousands of different antigens, but a given antibody will only be able to bind one specific antigen, okay? Because this is our specific immune response. But no matter which type of antibody the B cell releases, they're all going after that same target. So if that initial B cell detected and destroyed and presented an E. coli antigen, then all of the antibodies made by that B cell are all going to be going after E. coli. And as you can see down here, you don't need to memorize these numbers, but an antibody can produce 2,000 antibodies in a given second. And all 2,000 of those antibodies would be going after and hunting down E. coli. Okay? But a different B cell, maybe one that recognizes Salmonella, it would be only producing antibodies against Salmonella. Because remember, we're always looking for one specific target. All right, that wraps up this video. Let me know if you have any questions.